Hey, everybody, welcome to yet another journal club brought to you by Life Extension Advocacy Foundation, Lifespan Extension Advocacy Foundation, lifespan.io. So I'm here with the usual group of folks that are joining me every month. This is end of March here in New York City, where it is unseasonably cold. And right now we're going to be discussing a paper on epigenetic reprogramming in vivo in mice. From the Belmonte lab and various collaborators, Horvath of the Horvath clock fame, and others, basically from universities spanning across Spain to the West Coast of the United States. And I don't recall, Steve, if we do the paper with the Belmonte paper with the lamin A, where they basically got lifespan extension with mice. Or did we skip over that one? This builds on this work.
I don't think we've done that one, but we did the very old one. Well, so very old, from the end of 2016, where Ocampo and Belmont demonstrate that it was possible to reprogram living mice, but they were specially engineered, if you recall. So this is a little bit more of an incremental step forward, but I'm not going to spoil it. No spoilers.
Sure. So this builds on previous work that Belmonte and colleagues have done. I believe the last paper which they mentioned in this paper was the extension of mice that basically had a laminopathy, a lamin A mutation. Hutchinson-Gilford progeria is the human condition which basically leads to presumably accelerated aging phenotype. And they showed that basically reprogramming using these OSKM factors that are also known as the Yamanaka factors, you can get at least partial rejuvenation of tissues and an extended lifespan in these mice. And this was their previous paper. Of course, these were hyper accelerated aging mice. These were not normal aging mice. So they take this work a step further, where they look at the phenotype of mice. I believe the mice they use in this paper are C57 black 6 mice. And another group of C57 black 6 mice that are been genetically modified to have these OSKM factors, which are transcriptional factors that are involved in cellular reprogramming, so changing cells into induced pluripotent stem cells. And these so-called Yamanaka factors are under the control of a doxycycline-inducible promoter system. So these are the mice. So let's share the screen here, take a look at the paper. So Steve Horvath, not John Horvath, and Belmonte and Jasper. And so this is the Browder et al. Paper. And just move this little bubble out of the way. The title is In Vivo Partial Reprogramming Alters Age-Associated Molecular Changes During Physiological Aging in Mice, right? So this is during normal aging in mice. The only thing abnormal about these mice is that they have been modified through a transgene to express these Yamanaka factors. So these factors, OCT4, SOX2, KLF4, and c-MYC. There are a couple of other ones that were initially involved in the earlier papers, but these basically are involved in, changing cells to a stem cell, more youthful state. And they do that by basically altering the epigenome. And one thing that they look at in this paper are various epigenomic modifications, hence Horvath is on the paper, that relate to the methylation status of the cells, right? So we do know that the methylation status changes, and that this could be used as a quote unquote clock, if you will, to figure out the biological age of an organism. So let's take a look at the data here. So figure 1. So there's a couple of crucial things to keep in mind. When previous studies have been done with these OSKM mice, they used kind of a long-term induction, and from an early age, I believe, I think these were constitutively, transgenic mice that were basically producing OSKM factors. And these mice had problems. You had teratomas forming, so you had tumors and cancers. So these reprogramming factors, you have to dose them very carefully. It's not like a kind of taking a supplement where certain supplements, it's not too much of a problem, taking little excess NMN or nicotinamide riboside. These are pretty potent factors. They'll reprogram the genome of cells, and basically you can end up with cancers if you're not careful. So they did a slightly modified regimen here, and in Figure 1A, they basically, I believe if I'm reading this paper correctly, I read it a few times over, they have this induction cycle where so these mice it's an inducible, so this is the genotype. You can get these from Jackson Laboratories, I believe, R26, RTTA heterozygous. So these are these heterozygous mice. So there's one copy, I believe, of these OSKM factors on one chromosome. This locus is inducible by giving doxycycline, I believe, probably to the water supply of the mice. And I believe it's also dosed periodically. So there's an induction cycle where they give doxycycline for 2 days, and then they wean the mice off of doxy for 7 days. And they do that, if I'm not mistaken, for the duration of the treatment. So they have a whole bunch of these mice that they use as comparisons, various genotypes. The 4F is the mice that have been modified, the transgenic mice, and the B6 is the normal wild type genotype, the background of the mice. And they look at various ages. So 3 months is a youthful mouse, and then the older mice are 26 months, 22 months, and 25 months. And this relates to the dosing regimen, right? So treatment duration. So basically, this is the starting age. This is when they basically do the doxycycline induction regimen. So these mice are not, they don't have their OSKM factors switched on from the beginning, from the get-go, which, there were issues of severe toxicity with this. They do it much later in the mouse lifespan. So starting age 12 months, and the treatment duration is 10 months, and then the takedown age is 22 months. So that's when they sacrifice the mice and do various analyses. Treatment duration in this mouse here was for 15 months. So, 7 months. So it was started a little later, 22 months. And then they had a very short-term duration where the mouse was already old, 25 months, and 1 month treatment duration, and the takedown age of 26 months, and then something a little bit more kind of intermediate. So they had various kind of dosing schedules and regimens. So the treatment duration basically lasted throughout either late in the mouse's life, or kind of starting more in middle age and going to this point where the analysis was done. And then the authors looked at a number of different parameters. They looked at the DNA methylation status of these mice. They also did RNA-seq analysis to see what happened to the expression profiles of the genomes of these mice. They did mass spectroscopy, I believe, from the serum of the mice to look at metabolomics and lipidomics, looking at small metabolites and various lipid compositions between old and young mice, dosed versus undosed, to see what the changes are and if they correlate to a more youthful profile. And then they did histological staining and a wound healing assay for the skin of the mice, right? And these are the various tissues that they looked at for the various omics assays, and the actual, I think the healing, the wound assay was done only in skin. It wasn't done in any of the other tissues. So I'm going to stop sharing and just pause here, have a sip of coffee, and let that settle in. That's the protocol, more or less. And I wonder if anybody has any comments thus far. Probably not, since we just kind of laid the groundwork here.
I'll throw in one comment. So I think before this, I'm trying to think what the longest partial reprogramming duration has been before this paper. Because this 10-month treatment, I'm pretty sure, is the longest anyone's done, which is one of the highlights of how they sell this paper. I'm not really sure what the second longest was. I think at this journal club we might have covered a paper that did 4-week follow-up.
Yeah,
but I think that was the best around until this thing was published.
Yeah, it'd be interesting to see if at this point there's probably could be a good review paper that does comparisons of all of the different OSKM dosing regimens. If such review paper doesn't exist, right? Somebody could write one, because it could be yeah, I think this might be the longest, albeit the longest starting later. So it's a it's a conditional induction like later in the life, versus starting early, where I think that was where they had problems in vivo when they started this like early kind of youthful age. But then again, they also have this pulsed kind of dosing. So I think this, even though this being longer, the dosages might be again, I don't have, probably very difficult to compare between different mouse genotypes. The expression levels of the various OSKM factors between, the dosing regimen here versus another mouse. So this is longer, but the dosages might be lower. Although I might be talking out of my ear here because I don't have a paper to compare it with. So the non-conditional expression of OSKM may have been problematic because it was started earlier, but also could have been problematic because they just had higher expression levels of OSKM. It'd be worth looking into that for those of you who are interested in dosing, although a little premature right now to extrapolate from the mice and try to dose yourself intravenously with OSKM factors. Probably not recommended.
A question that arises that is not covered in the paper is, how can people do this if we're not genetically engineered to have this extra 4 genes put into our DNA? Do you have any speculations?
Yeah. Well, what was that other paper that we covered? Basically, let's take a look at the current vaccine technology using, I can imagine using, either using a modified virus or liposomes, right? That you might have your messenger RNA that encodes these OSKMs, right? That are just sort of incorporated either intravenously or through some other method into your cells. Now, of course, that's got to get into most of your cells, although probably not. It could, a partial improvement might be a good enough improvement, right? Like we don't, knocking off a significant amount of physiologic age from a couple of organs is still pretty damn good versus, rejuvenating the entire body back to a very youthful state. So, the previous paper that we covered, I believe I was pretty excited about it because it had a really cool combination of technologies, right? So they basically made CAR T cells in vivo. I think that might have been the first time CAR T cells in vivo using a liposomal approach that was not unlike the approach that's being used right now with the current round of Moderna and Pfizer vaccines, right? So a liposome that had messenger RNA encapsulated that expressed a chimeric CAR T receptor. And that was a pretty cool technology because number one, it was done in situ, in vivo in the organism, and also it's a lot cheaper, right, than extracting the cells from a patient and modifying them and putting them back again. So that platform could be applicable to all sorts of other types of modifications that you don't want to have be permanent, since this is an mRNA type based technique. So an OSKM type partial reprogramming using this type of methodology, might be a good kind of, I don't know if anybody in the Belmonte lab is looking at this, but might be a very nifty, next step, if you will, to see if, to go from a transgenic, like you mentioned, John, to something that could be more usable as a therapeutic, right? So yeah, I'm with you on that. So that would be, that would be my thought as far as a tool, because yeah, the main, the transgenic mice are there to see what happens, but the actual that you're not gonna use a transgenic approach, at least at least for the approach that's used in the exact transgenic approach used here, is not going to be translatable to to a therapeutic application. So, and there might be other delivery methods in the wings right now, but that was sort of the state of the art that we covered a few months ago. So if anything else is better, I'm all ears, and we'll do a journal club on it. Yeah, great question. So let's go back to sharing the screen. Whoops, let's see. Let me just one moment. Every time I close this paper, okay, now I go open this. Sometimes share disappears when I just had. Okay, so so this first figure is important with regards to safety, and that I would posit that this is unlike a lot of other papers where you have a lot of background information for how they generated the mice and made sure that things are expressing. It's sort of like you could kind of shove it into supplemental, but this deserves to be prominently put up here because again, OSKM factors, they can cause cancers and other problems. So they went out of their way to show that this was safe for these mice. And by safe, I mean they look at certain characteristics of the mice. They look at body weight. So minus doxycycline n equals 11 plus doxycycline n equals 10. These are 4F mice going from 15 months where you had the dosing regimen starting, so 7 months of treatment duration until 22. Body weight seems to be okay. The survival probability, we'll get to that in a moment. Again, no statistically significant difference. They looked at white blood cell levels, neutrophils, lymphocytes, monocytes, and they couldn't get a difference in hemoglobin, so various blood factors. And also, and I'll make a comment here in a few moments. Also, they looked at various performance characteristics. So figure E is, let's see, what is this measure? 22 months of age, 7 months of treatment starting at 15 months of age. I don't know if they might have in the material in the extended data, the other dosage regimens. I can't recall, but this is the one that they have presented here after 7 months of treatment. So the distance traveled, the number of vertical events, which is probably a mouse climbing up the bars here, average velocity of the mice was not affected. So there appears to be no difference. The only thing I would mention here is that that's just maybe somebody else can comment. That's great that there's no difference, but I would think, shouldn't there perhaps be an improvement? Because right, these are older mice. So it's good that there's no difference. Good that there's no negative consequences, right? But I don't know. I would think that if you're looking at various performance characteristics of the mice as well, right? So like crawling around, jumping, and if you're measuring that, and that's something they don't do for the rest of the mice as well. So I would say that if I would say a good follow-up to this paper would be looking at later age physiologic parameters dealing with how the mice actually perform, right? So if the mice are actually exhibiting youthful vigor, right, they would perform better in a Morris water maze test. They would run faster. Their circadian rhythms would be not as aberrant as an older mouse because mice are nocturnal. So so you could measure these things by having a webcam and just and looking at how these critters perform, right? So and then of course the question always at the end is well, do they live longer, right? Unfortunately, we can't tell right now because the mice were sacrificed at those points to basically do the analysis of the methylation status and the lipidomics and the metabolomics and the other stuff, right? So so we'll get to that in a moment, and it does appear to be an improved profile. So right now, right off the bat, safety looks fine from the parameters that were measured here. But I'm just wondering, if the mice if there is an improvement, is it because the mice were sacrificed early and the improvement wasn't measured later, or is it or do the OSKM factors need to go a little bit higher? Because we'll see in a moment the expression of these OSKM factors might not be high enough, because it varies between tissue to tissue, and the methylation profile kind of suggests that. So anyway.
So I was actually really happy with their safety figure. Like, what your points are, I think, well taken. Like, where are the improvements at? Yeah. But there's a lot of stuff with this treatment that I feel is kind of should have been wildly dangerous. Like, they apply their inducible OSKM construct to the whole mouse, like all the tissues, and there's no tissue specific dosage. It's one size fits all. They do the whole 4 OSKM, whereas like other papers have tried to dial things back. There's been reprogramming as OSK with the idea to make it safer. So if someone had pitched this study to me, I would expect what you were describing earlier. Like, you're probably going to give your mice cancer. They're probably going to have terrible things happen to them, and that doesn't happen. They get away with it, and that alone, I think, is a really impressive finding. They are probably, I'm assuming they're already doing a follow-up study to like try to dial it up.
Get up, yep.
But that they could do this at all, I would not have predicted, and it's kind of cool.
Yeah. So definitely the safety stuff that, like I said, deserve to be in the forefront there, and again, there's a ton of work here, so it's not like I'm not playing armchair quarterback here and saying, well, they should have had an improvement. These mice should have been like super mice. What's going on here? Well, I appreciate the amount of work there, and like you said, erring on the side of caution. Like we sort of have a rough bookend, because these are very painstaking trials, right? And if we can go back and compare what's too much and what's too little, right? You can start kind of getting to this point where you can start getting real differences. So this is more at the kind of the safety end, and albeit you do have expression of these factors throughout the tissues, started later in life and pulsed, versus something that's turned on early and probably at a very high level and leading to cancers and other problems, right? So here we have an approach that's not causing cancer, at least in these mice that they've looked at, this one genetic background. So, and that's it. And that's a good thing. And they do see some modest improvements. So, let's go back to paper. Okay. So that was figure 1. And figure 2 is a methylation clock analysis. And a lot of these analyses, I kind of sort of joke sometimes that these volcano plots and these gene expression analyses, a lot of this data isn't meant for human eyes. So there, wow, 1,507 things different, but you have to kind of go through and figure out what they are, right? So what you can tell from these plots is that things kind of shift back towards a more youthful state, and then you have to tease through, what exactly is shifting back towards more youthful state when it comes to the metabolites, the various lipid profiles, and so on and so forth. So anyway, they use this clock here. So I'm not familiar with too many of these clocks. What's it called? What's the L stand for? U is for Uber correlation. I know Lucas stands for last universal common ancestor, but we're not doing evolutionary biology here.
I think it's just lifespan Uber correlation.
Yeah, I think so. Anyway, so that's, so there's various types of these clocks, and I believe these are the, the p-values up here. So you can kind of look through here, and we won't go through all of these individually. And this is age association. B6 is with doxycycline, B6 plus doxycycline. So that's a control here. So it's good that they're controlling for any changes of methylation that might happen by dosing these mice with doxycycline, versus 4F plus doxycycline, which is the mice that have the transgene, right? So these are these mice that have been dosed starting at 15 months, so 7-month treatment. And they look at the methylation status. And really, I'll just kind of zoom in on some of these things, just the two that are significant. The only real changes that they see are in the kidney clock, so the kidneys and the skin, right? So the kidneys and the skin of the mice. And they don't really see much of a change that's statistically significant in muscle or spleen or lung or liver or any of the other marks. And they don't see as much of a difference also in the kidney clock and the skin when you do this analysis, when you do this dosage much later for a very short duration. So these mice are the 25-month mice that have been dosed for 1 month, right, and then sacrificed. So they mentioned that in the paper. So again, kind of narrowing down into kind of a therapeutic window here, right? So you know, starting this particular regimen too late, you know, for too short is not really effectual. Starting it, you know, earlier, at least for the 7-month duration here, you do get changes that are measurable. And the first measurable changes they have here that they look at is with these methylation clocks. But again, it's only for skin and kidney. And they do a wound healing assay. And one thing, you know, again, I'm thinking about other physiologic tests that can be done. So if it's kidney function that's improved, and it's, you know, at least the methylation status suggests that way, you know, again, just throwing out follow-up experiments here. I wonder if the renal function of the kidneys is improved, right, as well. Are the kidneys filtering, you know, as good as they could? I know they do some analyses. They do some tests with kidneys when they add a toxic substance, and if the kidney is able to repair itself, then, you know, then it has a better. And I think we did that in an earlier paper. I can't recall which one, but there was a test that was done, some sort of a nephrotic toxin, I believe. And also for the skin test, right? Do the mice? I'm just wondering, do the mice look younger? I don't see any pictures here. Old mice kind of do look old, right? They have look like they have scoliosis and their fur sticking out all over the place. And they do when they do the genetic analysis here, the gene expression analysis, they mentioned that the genes involved in hair follicles, hair follicle development, and other skin associated genes are shifted into a more youthful state. So I don't recall reading in the paper if the mice had a more silky, lustrous sheen of hair, right? I don't know. You know, they do better, I think, you know, in the wound healing type assays that they look at later. So I'd be curious to see how these mice look like. But suffice it to say, for these methylation clocks, the most robust changes that were observed were kidney and skin, and nothing really else significantly. There might have been some trends. So that kind of suggests, if we look at it, that, you know, that maybe upping the dose and doing it longer, you know, might actually shift, you know, these methylation clocks for other
To the author's credit, they're totally open about this. But if you go down to their extended figure 6, they have kidney and skin specifically with some of the other clocks. I think they're all previously published Steve Horvath clocks. And the significance varies. And sometimes the effect is reversed. Like, panel B, kidney clock, the center panel.
Yep.
That's a big effect, and it's the wrong direction.
Right.
Or it's not the desired direction. And I don't know which clock is the right clock. I'm not sure if anyone's really sure, like, what's the best way to be assessing this, but the results definitely vary by the methodology.
Well, like I said, one way to kind of tease that apart is to do an actual physiologic analysis of the kidney function, right?
That is kind of the final word.
Right. It's like if it's like, okay, which clock holds? Do the kidneys perform better or crappier? If they perform crappier, we know we are kind of you know, nudged in the other direction there, right? So you know, that is kind of the final. But yeah, thanks for pointing that out. There is variance with these clocks here, and they do show that data. So the other thing to point out is that there's go through all of this, but where's the other extended data? So, they do expression of reprogramming factors in tissues, right? Relative levels and versus GAP-DH, which is, your general housekeeping type of gene that they look at. So these are these OCT4, SOX2, KLF4, and c-MYC in 9 months after 48 hours of doxycycline treatment. So I don't know how, again, it's kind of hard to correlate that because this is in 9 months after 48 hours of doxy treatment, right? So I don't know how these would vary throughout the course and duration of the treatment, right? If you started earlier in the mouse versus later in the mouse. And I believe that these are, 4 different mice. N equals, two are dox without dox, and then 3 are with dox. And there's quite a bit of variability between these mice, right? Like, like these 3 mice here, the OCT4 levels are, it's like a twofold increase difference between some of them, and depending on the tissue. So kidney, let's see, where's skin here? You have skin? They don't have skin? Oh, yeah, there it is, skin. I don't know why in the dox, is that dox minus? The dox minus cells, mice that you have this huge level of KLF4 in skin. So, I mean, there is also variability, right, with this expression. So, I don't know, when you do these types of tests and you aggregate the results from all these mice, right? Could you do I don't know if they did this type of correlation in this paper where they matched the levels of expression of these OSCAM factors between each mouse with epigenetic status, the methylation status and other things, or as everything, as shown in the figures, everything is just aggregated together and you've got your error bar, right? But is there a correlation going on between those levels and what we're seeing with any variability with the data out there? Because there is variability in the expression, quite a lot, in at least those 3 mice that they showed there.
It's not just mouse to mouse either. Like, it's also gene to gene, which I have a hard time comprehending. So their construct's polycystron, right? So like all 4 genes should be on the same transcript. Mm-hmm. They get different amounts of their different genes in the same mice.
That's right. It is a polycistronic. It is one transcript. Yeah.
And like there's lots of, like, there's a million molecular biology mechanisms that could like regulate genes to cause differential expression. Mm-hmm. But like if you're the authors and you're thinking about how do I dose this better? Like how do I control the amounts of these factors in a way where I can like bump up the effect? Mm-hmm. I think it's kind of unnerving that there's both of these types of variability, because where is this coming from and what can you do about it?
Yeah, right. You would, like you said, you would think everything coming from like one location, transgenic mouse, one very homogeneous genotype, C57 black 6. Never mind doing this on different patients, right? Wildly different genotypes, but also coming from one genetic locus that in of itself is a polycistronic locus coming from one messenger RNA transcript, right? So to their credit, I mean, this is trying to get as controlled as possible for the expression, and yet you've got this variability, right, happening between different mice that are ostensibly clonal, right, for what's happening, and at the same exact ages. So, and between different genes as well, right, within the same mouse, between the 4 different factors. So, yeah, I don't know what could account for that. Environmental or some other subtle genetic differences. It could be just the amount of doxycycline these mice are drinking. I mean, unless it was done by gavage or for maybe that's what they did. I have to double check that, because for the long-term dosing, they probably just fed it, but here they might have done, for consistency's sake, oral gavage. So which means they squirt it down the mouse's throat. They could be a different uptake of doxycycline. Don't know, but there is that variability. which, you know, goes to show, just again, another caveat for that they managed to get a good safety profile despite this. But translating this to humans, where everybody's genotypes are much more divergent, not to mention the environments everybody's in, you know, these factors have to be most likely kept within a very tight window of operation, right? So that, you know, a lot more work needs to be done to make sure that everything is in that window. Okay. Okay, so here they have a lot of omics data. So figure 3, I'll come back to it. Figure 4, figure 5, we'll skip over. That's not omics data. That's you know, they noticed changes. They noticed a lot of the omics data suggesting that you have, you know, skin, like I said, you know, more youthful, and also kidney. So they do a wound healing assay, and then figure 6 as well. So where they do metabolite and lipid analysis. So let's see what they do here. They do a gene expression analysis, transcriptional profiling. Yeah. And RNA-seq data. So let's see, what's the most important thing that we can get out of this? This is where they notice the enrichment of certain genes. So, you know, they do these volcano plots, which basically tell you a difference in, so anything above here, I believe, is a p-value of less than 0.05. So these are different genes that are either upregulated or downregulated significantly. These are 26, what is this, 26 months, so old mice without doxycycline. So this is your control, old mice and young mice, right? So this is the difference in the RNA sequence profiles, and that profile changes. There's less of these There's, you know, less of a difference in the amount of RNA expression changes that happen in 26-month-old mice that have been treated with doxycycline. So this is actually only 1 month treatment, actually, right? So there's like a slight difference versus 3 months minus doxycycline. So it appears to be a slight shift. I think in the paper, they mentioned that it's not that significant, but you know, there is an improvement. I mean, in that, you know, that you have less of a difference. And when they look at the different, you know, top downregulated and top upregulated, I think later that a lot of the ones that are downregulated, you have less, you know, less senescence associated factors. But here's a whole bunch of cells that are genes that are upregulated, that keratinocyte migration, hair cycle, intermediate filaments, right, which connect cells together. So a lot of these changes that they then explore later. So, you know, the arrow pointing to what these changes correlate with: motility, migration, hair development cycles. So that basically enabled them to, you know, work on a wound healing assay for figure 5, I believe. You know, at least point to that. They use that data to help kind of, you know, sculpt one of their physiologic tests, which is, okay, if the skin is improving, then let's do an assay that basically see if we actually have an improvement in skin function. And one type of assay would be a wound healing type assay. Although I'm sure there's other types of assays, like a skin elasticity assay. So there's lots of other types of things that could be done to see if there's an improvement. But they went with a wound healing assay. So any questions? A lot of omics data. Go back here. So
it looks like they also measured skin thickness.
Yeah, I think, yeah, they looked at, they definitely looked at collagen deposition later. Did they measure, was that in figure 5, or did they mention that the skin thickness in
it looked like it was in one of the figures where they would show untreated and treated mice. Also, yeah. I believe the doxycycline was administered in the drinking water according to the methods section at the end.
Gotcha. Yeah, I mean, that would be a lot of variability. I'm sure, you know, every mouse has got its own thirst response, right? So yeah, I don't know if it's possible, but I don't think you can. Can you measure doxycycline? I mean, you could potentially measure doxycycline levels in the serum of the mice, but we're getting to a point where that's a lot of work, right? Measuring, taking blood draws and measuring dox levels in the mice. Well, I guess you could do that at the same time as you do the other serum analysis, but I'm not sure.
I'm guessing everybody drinks about the same amount of water because they don't have anything else to drink.
Perhaps, yeah, within a range. Okay, so let's see, what did we do here? So long-term reprogramming restores metabolic gene expression and reduces, as I mentioned before, inflammatory SASP gene expression. So we have a lot of, well, again, these are gene expression, analyses that have been done in this case, B6 mice that are untreated and 4F mice that have been treated for, was it 15 months? So 7 months again, right? So 7 months treatment. Basically we have, the most major changes have been pointed out here in figure F and also at E, where you look at essentially, minus doxycycline and plus doxycycline, and you see basically a down regulation of, inflammatory factors, which are correlated with aging, senescent SASP factors, and that's good. Sure, there could be a lot more follow-up analyses that could be done, but I think one attribute of this paper is that you have just this massive amount of kind of like very raw data that's presented here that other researchers as well as these researchers could look at and say, gee, let's see if we can follow up on this, right? Because now we could see that what are all these factors that are going down as well as up, and how can we follow up with this, right? Because if we go to, I'm going to skip figure 5 and come back to it later, but if we go to figure 6, you have a lot more of these assays here, and these are polar metabolites, right? So polar metabolites are things like ATP, cyclic AMP, things like that. I don't know if they looked at those specifically, but malate, urea, uracil, creatine, and versus lipids, right? So they did basically mass spectroscopy from blood serum, and they looked at all of these different metabolites. And the bottom line is that a lot of these metabolic profiles, and all of these metabolites, we're not going to go through these, because if you ever opened up one of those biochemistry charts that come with like from, like usually free from like from Sigma that covers up like half your wall, you can see that there's, the Krebs cycle is easy, right? And then you got about 5,000 other pathways that connect to it. So a lot of these metabolites are involved in all of these pathways, these biosynthetic, these, anabolic and catabolic pathways. And so what a particular metabolite means, I'm not going to go into, right? Well, like why is it that a certain metabolite goes up or down in a young mouse, right? So that's going to tie into a very, a particular biochemical state that I would assume is more efficient and more optimal in a younger mouse, and that's why you have that level of a certain metabolite versus a difference in an older mouse. So now we have 17-month-old, 22-month-old, and 3-month-old mice. So these are the young mice right here, and no doxycycline. So this would be like your positive control. And then we have your plus dox and minus dox, plus dox, minus dox, 17-month-old and 22-month-old mice. So what is this? 22-month is 7-month duration. So how long do they dose this? Okay, so yeah, so 5 months versus yeah, so they have 2 different durations here, and they look at the profiles of these compounds, and let's see, by eye, does it shift? Well, it sort of looks like when you look at plus dox, you can see some of these lipids here, these lipid profiles start shifting towards that of a more youthful state, right? And they break this down for some of these compounds here. I don't know if anybody here has a particular favorite compound that they can talk about, such as 2-hydroxy 3-methyl valeric acid.
So there's a few.
Yeah.
So in the text, I don't think the authors try to explain all of these, but they mention that they get a lot of hits for one carbon metabolism. And specifically, there's some of the ones in the middle of this chart. So it's the dimethyl and trimethyl lysine, and the SAM, the S-adenosylmethionine. So for me, at least, those are really interesting hits because earlier we were talking about, well, what's exactly going on with their epigenetic clocks. This one carbon metabolism stuff is really important for epigenetic changes, like the di and trimethyl lysine. These are things that are going to be like inextricably involved with histone methylation status. And the S-adenosylmethionine is like required for any sort of DNA, like CpG methylation. So when I see increased levels of those metabolites, that seems like pretty good circumstantial evidence that something's fairly substantial is happening on the epigenetic level, even if it's not really clear what's happening by the clocks.
So here we could see, so the p-value here, well, they say it's 0.12, but you could definitely see a trend here, right? So this is going from, and trimethyl lysine, the levels are low here with 3 months, no doxycycline. They go higher, 17, 22, then green is 17 months plus doxycycline, and then 22 months, right? So you definitely see these trends here where you see a recalibration back to a kind of a more youthful state for some of these metabolites, like the one that you mentioned, and trimethyl lysine. So there's a lot of this data, and also with regards to lipids, I believe SM is sphingomyelin, PCF, probably phosphatidylcholine, right? So these are important lipids that are found in plasma membranes. Okay, so besides the health data that they did earlier, the other physiologic test that they actually do on these mice that looks like there's some changes correlating with improvement, and these are some of the major changes, the most changes that they saw, at least with one type of methylation clock, was pointing towards the skin. They basically, they look at skin in response to injury. So 4F mice that have been sacrificed at 22 months, so 7 months of treatment. Then they do a skin injury, or they do a skin injury at 22 months. They doxycycline off. So they stop dosing with doxycycline during this 14-day injury, to make sure that there's no confounding influences of the OSKM, right, being turned on at the time, because that makes sense. It induces pluripotency, so they want to make sure that that's kind of a good thing to do there. Make sure that at least during this window here, when the OSKM levels are low, that it's because of whatever intrinsically has already happened to the tissues that we're observing here. So they do this. So this is a Ki67, I believe it's a proliferative marker. And these are some immunohistochemistry of tissue cross sections of the skin, young, old doxycycline, and old plus doxycycline. And they measure the amount of these cells that have this proliferative marker, which I'm assuming it's these brown spots, and per micron. And they note that basically you have more of them, and it's significant between young and plus doxycycline versus minus doxycycline and plus doxycycline, right? So, it's an improvement that shifts it towards the young state. Now, it's kind of, I don't know, when you look at the young, like this one thing that's not mentioned, I mean, qualitatively, right? Well, I guess you can quantitate it too. You do see a lot more proliferation at this zone. I'm not sure what, that's the epidermal layer, versus here you have a lot. It seems like you have a lot more interspersed in deeper layers as well. So I'm not sure if, because when they do the methylation state, they look at skin cells, right? But you have different, you have skin and different layers here, right? So, and this far down, I'm not a histopathologist. I don't know what we're getting into here. More connective tissue also, perhaps, right? Versus more, epidermal tissue up here. So it's anyway, that's just something I was like staring at this. I'm saying, okay, yeah, per micron, right? But is that yeah, it appears that there might be, I don't know what that means, that you have less, or it appears certainly to my eyes that you have less deeper down. Although with the old with minus doxycycline, it just looks like there's less proliferating cells in general throughout this entire throughout this entire skin. And I think, John, you mentioned that they talked about thickness here as well. And I was at figure B, because it does look like you can see difference of thickness also up here in the KI67 slide, but figure B, histologic wound area by hematoxylin and eosin staining. And that's just the area that they wounded. And they measured fibrotic tissue with this serious red stain that basically stained for collagen deposition. So they mentioned that during this wound healing assay, you had less fibrotic tissue forming within a certain given area. That's, more akin to the young mouse, right? I'm gonna stop sharing here. So there's a lot more buried in this paper under the hood, so to speak, in the extended data. There's like 10 figures. So they give a lot of background there. And if folks are interested really in designing experiments around these OSKM factors in mice, it would be good to dive into that data, because that's where you can see all the differences and the nuances of expression. Certainly the differences in the methylation clocks, as Michael pointed out, and also, as I mentioned, just the expression of these OSKM factors between the different cells, right? So despite all of that variability, there does seem to be a measurable improvement in these mice. In vivo partial reprogramming using this technique doesn't appear to be toxic to the mice, which is a huge plus, because these are, normal mice, or at least as normal as a lab mouse can be in a transgenic background. And you do get slight measurable improvements in the, significant improvements, in various metabolites, and also, methylation status in a few cells, and also improvements in at least one major organ, which is the skin. So it would be interesting to see, again, in the follow-up experiments, there's a lot of follow-up experiments that could be done here, which is also the mark of a good paper, because it points you into more experiments. But, different dosing regimens, certainly, that's a big one. Different methods of inducing the OSKM factors that are more translational, right? Such as using some sort of a virus or a liposomal technique. See if there could be more improved methods to look, to control for the variability of these factors. I don't really know how. You could certainly, for the purposes of an experiment, do this on 1,000 mice and basically just correlate the data with the mice that have the closest expression profiles, right? And try to get as little error bars as possible using that approach. But then again, in practice, what accounts for all that variability, and can you tightly control that? I'm kind of at a loss right now as far as thinking about a way to do that. And of course, other physiologic tests that would be great to see, such as looking at renal function, right? If you have an improvement, different types of studies on the skin, and of course the big one, increased lifespan, right? So I think that we might want to see an increased dosage. I'm thinking that if you have just these changes in some tissues right now, probably the dosage has to be boosted, albeit carefully, before we see anything that is as powerful as an increase in lifespan. Now they did load, again, this is kind of relative, but they did, they pulsed the dosages of OSKM for kind of halfway through the the midlife of the mice, and they said that this regimen didn't seem to be so significant if you did it at the last month of the mouse. I'm wondering if they tried, or somebody else has tried, like a high dose later on in the mouse's life cycle, right? Like seeing if you can if that would if that would still have a problem with regards to teratomas and cancers if you did it really late in the mouse, in the mouse lifespan. I'm not sure, but certainly I think there's a lot of room still to figure out the proper dosing regimen here.
Well, I'm wondering how you could change the dose, because they've got this cassette in their genome. And no matter how much doxycycline they drink in the drinking water, it's not going to make a bigger dose.
Yeah. Yeah. Well, not these mice in particular, but they would have to, well, that being said, I believe these are heterozygous mice. Could always do homozygous mice, right? So I'm not sure. I'm not sure why they didn't. Let's see, let me go back to this paper, because I'm pretty sure it was a heterozygous mouse, right?
Well, you could also give different mice multiple cassettes. So drinking the doxycycline would turn on more genes.
Yep. You could certainly do that. You could have homozygous mice. You could add in more cassettes, right? So you could definitely trying to think here. Trying to think also if another technique to sort of like that would prevent the mice from having a massive eruption of tumors all over the place. If you can have multiple cassettes in one particular organ, right? Because if you have a promoter that's basically expressing this only in the kidney, and then you could wildly fluctuate the dosages. So if something crazy happens, like it's going to be isolated to kind of the one organ, right? There might be less like initial lethality, and you could modulate the dosages much more, much more, and the mouse might be able to tolerate it more, and then you could do more kind of analyses that way, versus having it throughout every single cell of the body, which is this study right now. So that's possible. And it could be that different tissues might need different amounts of OSKM factors, right? So you might need higher dose, again, I'm thinking out loud here, you might need higher dosages in the spleen or the liver versus the skin, right? So without looking at the expression right now, I'm just going to assume, and I might be wrongly assuming, of course I'm wrongly assuming here because we saw the expression fluctuating all over the place, but, assuming that the expression is the same in liver as it is in skin, why, then you would expect you have a more youthful methylation status in both, but you don't, well, at least with one type of clock, right? So maybe you need a different dosage of OSKM factors in the spleen. And again, this is, to kind of throw in another bit of variable here, organs, I believe, also can age at different rates, depending on also environmental stresses coming in from the environment, organs that are exposed, although these mice have been reared in cages. So you would think that it's, things are kind of consistent, but still, you can have variability between skin aging and kidney aging, right? So is there a finer, more sophisticated way to regulate the OSKM factors, right? You might need to regulate different levels in different tissues at different times to take into consideration those differences. And in this case, as was mentioned, you're expressing these factors at lower levels, but the attempt is to make them as consistent as possible at the same time for all organs, right? And
if follow-on experiments switch to the idea that you suggested of injecting mRNA like a Moderna vaccine, this might give us more control over which tissues accept this this vaccine. If we could put some sort of targeting on the vaccine, and it wouldn't be a vaccine, of course. I'm just using that word because it's analogous to Moderna.
Yeah. So that's definitely I think that's could be a worthy approach. Controlling, of course we'd have to control for like the uptake, but yeah, if you had liposomes with different different ligands that can bind to different tissues, you can then control the dosage to different tissues, right? So yeah, I think there's a lot of of variability here that you can mess around with to try to get to the bottom of the optimal dose, right? And again, the optimal dose might, when I say optimal, I mean truly optimal, might require, quite a bit of sophistication when it comes to different levels of these factors in different tissues, maybe even induced at different time points, right? And if there's, so right now that's sort of, well, it's definitely out of the not possible for current medical technology. It's something highly experimental right now, but that's something that could definitely be probed right now and looked at. So this paper here is a pretty good jumping off point for that. So it's a in that regard, it's a very pioneering paper, and I think we're gonna we've seen a couple of programming papers, and there's just going to be a lot more, obviously, after this one, that are just going to get, I think, more sophisticated when it comes to the levels of expressing these factors in different tissues. So that's my 2 cents, and yeah, I don't know, any other comments?
Oh, one of the authors, Belmonte, was in the El Pay, the newspaper that's very popular in Latin America, big newspaper, and he was saying that within 20 years, in his personal opinion, he thought that we could probably, I think he said, stop aging. I don't think you can stop aging per se. That's a strange turn of phrase, but he was talking about the potential of things like this. Partial cellular reprogramming with Yamanaka factors. Obviously, he's got a dog in the race here because he works at, I think he works at Altos Labs. Altos.
Yeah, just recently. But I think shifted his lab there.
But it's interesting that that he said, that sort of 20-year timeline, which is what we've been talking about quite a lot on the news outlet, and this sort of timeline comes up a lot with other researchers. So it's interesting. I think that's quite reasonable, 20 years for them to refine and perfect. What do you think about that timeline? I mean, do you think 20 years is reasonable?
Well, it's always dangerous to predict, right?
Oh, don't be boring.
Yeah. Okay.
Come on.
What was I going to say about this? Yeah, I was going to make a cheeky comment in that, aging has already been at the evolutionary level. Aging has already been defeated, right? Otherwise, the first cell that started to divide would have aged and sputtered out immediately. But no, things have been going along merrily for 3.8 plus billion years, and cells have mechanisms to basically get rid of the garbage, the entropy that accumulates, and there's a variety of mechanisms. And for actively dividing cells, right, it's asymmetric segregation of factors, and there's a whole bunch of other things that happen. And babies are born, reset to zero, yada yada. So aging in that regard, right, is the problem is solved. And again, I just want to stress here that the one thing that we're concerned about is really thwarting aging and increasing longevity in the evolutionary dead end that's known as our somatic tissues, right? So our somatic cells are not passing on our genes to anything else. So we're quite fond of our somatic tissue and all the memories that they hold. So we basically want to, a kind of, everything in biotechnology is kind of usurped from biology. So in that regard, I think, the same applies to longevity and anti-aging, right? We want to take all those mechanisms that, in a sense, at least, maybe there's some mechanisms that work for preserving the germline and having cells replicate indefinitely. We want that to happen to our somatic cells as well, right? So we want that type of rejuvenation to happen there as well. So the reprogramming that happens when a cell gets fertilized, it's something that people are trying right now with these OSKM factors and induced pluripotency. And then all the other problems that we have to figure out in terms of delivery to an already differentiated mass of trillions of cells, right? That we want to kind of all change. In 20 years, will we get, so it depends what you mean by that. I think definitely within 20 years, I think it's a fair bet to say that it'll be pretty groundbreaking, but I think we'll get at least partial rejuvenation of certain tissues. I think let's look at it from a medical perspective. I think some of these factors might be shown to, so right now we have a lot of people on transplant lists, for renal failure, for liver toxicity. I think it's possible, definitely. I think, the writing's on the wall that if you get the dosages right, and this reprogramming is actually very effective in situ, that we might, we might see I'll put my money on the table. We'll definitely see, rejuvenation of certain organs and having people taken off the organ donor list, and having those organs basically revert to a functional, if not completely, reverting an 80-year-old kidney to that of a 19-year-old, right? At least to a much earlier state where the organ is able to perform within a normal physiologic range, and that person doesn't have to have organ failure, doesn't have to have a transplant, right? And I'm just picking kidneys because probably kind of the vectors being used to target the tissues of that organ, certainly the liver, certainly other organs. But I don't really want to go beyond that for other tissues, as like the brain, for example. Because you have to get, if what you mean by we're going to have aging completely cured, I'm assuming what somebody has in mind, somebody who's 100 years old basically comes out of a pod and now looks like a 20-year-old, right? And in every sense of the word, performance characteristics, and has their memory intact, right? They have all their memories intact, right? That to me, I think there's a lot more questions to be answered, and there's a lot more questions that we don't know what those questions might be. For me to basically, say yes or no that that's going to be the state. So I'm going to say in 20 years, I'm going to be a pessimist and say no, that won't happen. But I will be an optimist and say yes, we will definitely have, given the technological landscape we have now and the way I'm seeing it develop, we'll definitely have in situ organ rejuvenation, and maybe moving the dial on a lot of organs at the same time, to a more youthful state. Perhaps not all of them, but quite a lot. Do I think physically we'll eventually get to a point where we'll come out of pods as 20 years old, right? We'll basically construct a techno fountain of youth. Yeah, I do. There's no laws of physics that are being broken here, right? I mean, as long as you just shove entropy out to the outside universe, the laws of thermodynamics are not broken, right? So it's an open system. So sure, of course, right? The Earth has been doing this for 3.8 billion years. Why can't our somatic tissues do the same trick, right? So it's basically it's not a. I don't think there's as long as laws of physics aren't being broken and laws of thermodynamics aren't being smashed, then yeah, of course. But I think the 20-year time frame, at least me personally. But then again, Belmonte's doing these experiments, so he has every right to be much more optimistic about this.
I think at least on the margin, this paper gives reason for some optimism. Like we were talking, you mentioned some of the authors on this paper, and they're big names. But there's also the point of where they're working right now, where pretty much all the authors here are currently associated either with Altos Labs or with Genentech. Which both have giant reserves and the ability to do a lot of research in the long term. And like if you're in the US and you're a biomedical researcher, your planning horizon is pretty much 5 years, because that's the maximum length of an NIH R1 grant. And it's really, really hard to guarantee funding on a horizon beyond that. And it's not private research is not all roses, but one potential advantage is they can think beyond the 5-term horizon if they have a dedicated research program. And another thing that made me very optimistic with this paper is, despite being a paper with a lot of corporate PIs, or at least recently corporate PIs, there's a lot of openness here. Like they report conflicting results, like with their clocks. They report a bunch of negative results. We didn't go into it, but to complement their skin healing study, they do a muscle healing study. And it doesn't work, but they talk about it. And normally, when you think about research coming out of corporations, you think, okay, they're going to hide anything that doesn't look good. Yeah. That actually did not happen here. And I was kind of impressed with the degree of transparency. So if they keep this up, I feel like whatever amount of research we can get done in 20 years, it's gonna be faster.
That's a good strategic point that you mentioned, that didn't occur to me, because again, academia. So I mean, there's problems everywhere, but and even the research I'm doing at a much smaller scale here at Leaf. I mean, the one thing that nobody wants to happen is to be scooped, right? Like you don't want to, but there's this yin and yang. You don't want to be scooped, but at the same time, you're also hoarding data from other people, because you want to wait until you're published, or wait for to do the analysis, right? And so you can get like the recognition, because that's sort of like the publications are really your currency, right, in academia. So I'm wondering if again, we'd have to interview the authors here, maybe interview like the executives at Altos and Jeff Bezos, right? Is there like a, or is the reason that they have this contradictory data and all this data and all this stuff being thrown out there is, in that is there now, at least maybe in this corporate setting, right? Is there obviously money being a big carrot, right? Is having less of a reliance on your publications or amount of publications, kind of being the sole or main arbiter of your worth as an academic, is that kind of dragging it all down? And do they have, do they have more flexibility in these settings where they're like, no, you guys have like a 15-year contract, 20-year contract, like doesn't matter if you publish one good paper or 2 good papers, you're locked in, we believe in you, here's tons of money, go for it. I don't know, is there some other influences, right? Because like you mentioned, Michael, there's a lot of big authors here, right? And they put out a lot of data, and even the negative data or the contradictory data is put in there. and it's there for people to look at and analyze. So I don't know. I mean, that's kind of a good, roundtable discussion type of, talk we could all have and invite other speakers, right, and see what the best way of doing science is.
Well, related to the 20-year question is, first of all, are you talking about 20 years till FDA approval, or are you talking about 20 years till success by an adventurous person or chimpanzee?
I'm viewing it as somebody coming out of a pod that's 20 years old, and I think regardless of where that happens, if people are convinced that this happened, I don't think the FDA is going to stop anything at that point.
I read about that pod in Bruce Sterling's science fiction book, Holy Fire. I don't know if you've read it or not.
No.
It was written in the 1990s, and it's amazingly prescient.
I got to check that out. So is that Bruce Sterling, you said?
Bruce Sterling, Holy Fire, and the Audible version is read very well as well.
Yeah, I read something. There was a similar. I got to meet one of my other favorite science fiction authors, Kim Stanley Robinson, who wrote the Mars trilogy. Yeah, he was the sci-fu, really cool guy. I got his autographed copy of his latest book. I think it was. I'm gonna mix up the years. I think it was the years like 2063. I think. And is the Red Mars. I think it was in the Red Mars of the trilogy. They discuss, the develop. There's actually a little section there where they talk about longevity technologies, because the people, the protagonists of the book end up living like into their 300s, and they talk about something not quite like a pod, but they basically have this regimen of like an intravenous system of different vectors that they put in, and they might actually use some sort of whole body electroporation technique or something. But it was using the book was written in the early 90s, the first one, or mid 90s. So they whatever the delivery systems were du jour at the time, that was used to get all these different treatments into the protagonists. I have to actually take a look at that book again, and I'm just curious to throughout, now that all these years have passed, what Kim Stanley Robinson had actually suggested. I wouldn't be surprised. At the time, right? It would be like antioxidants and probably telomere upregulation, right? So I don't know. I'm not sure what else would have been suggested in 1995.
Interesting. I'll take a look at that.
Yeah. So yeah, I think again, that's my take. So yeah, 100%, I think that we're going to, we're going to, if everybody's really serious about it, there is no reason why we can't reverse aging or enhance longevity, because you're certainly not, you're not breaking laws of thermodynamics, then why not, right? You just have to find the technological solutions. And it might be, and it's hard to, when I say it's hard to predict, it's because there's certain key discoveries have not been made yet, right? So, Yamanaka factors were discovered in 2006, right? Through kind of a very laborious, approach, by basically trying every single factor that they could, that they can try and winnow it down. So there might be other discovery, there might be other things that might pop up, and we realize that, there's a certain unique way that genes are regulated. I mean, I teach about CRISPR in my class, and if you talk to a biologist in 2010 and ask them, do bacteria have an acquired immune system? They would look at you like you're crazy, because all acquired immune systems rely on a multicellular organism. So it's like you're talking gibberish. That makes no sense what you just said, but yet they have an acquired immune system that's completely different than, that relies on a genomic locus and genes that basically target viruses. So there you go, right? So So nobody would have predicted that, right? It was a basic discovery, and now it's a technological tool that could actually be used in various longevity therapies. So you know, for that reason, you know, that's the only reason I'm kind of pessimistic about that 20-year frame. I am optimistic about having significant clinically translatable, you know, improvements in tissue and organ rejuvenation that could lead to getting rid of a lot of people off of the organ transplant list, you know, taking them off of those lists, so they can basically lead healthy lives and don't have to be on a dialysis equipment, for example, and so on and so forth. And I think that's going to be huge. And certainly that's sort of the, you know, if we talk about how these technologies phase into society, I think that's going to be, you know, that's certainly going to be a big one. And that's something that, at least in the United States, is you know, the FDA could easily approve, and will definitely, you know, be very interested in. And I think doing that for individual organs will actually even, you know, will help us get better data for safety to translate those results into other tissues as well. Because as we've seen with this paper here, you've got all this variability in a genetically homogeneous mouse, right? These are all clones. They're all generating, they're all in the same environment. And so same environment, same genetic background, a polycistronic inducible locus, and you've got these numbers fluctuating all over the place with like these, you know, these factors between tissues and in between mice. So you know, there's going to have to be a lot more tighter control over that, I think. So yeah. So going back to my prediction, yep, I'm hoping that there's going to be organ rejuvenation and people off the organ donor lists. But I'm gonna be the pessimist and say that I don't expect to see a 90-year-old turn into a 20-year-old in 20 years, outside of a magic act. But I you know, look, I might eat those words, and I'll be happy to. I just, you know,
maybe we'll see a 90-year-old turn into an 80-year-old as starters.
Yeah, that's different. That's different. So that's, you know, so I'm using an extreme, you know, so I don't know exactly, you know, what Belmonte and company were thinking, right, when they made that statement, you know, like a reversal of aging. So there's degrees of it, as we've seen right now. So I'm using the other extreme, that you have a complete resetting, right? It's like, basically it's like reborn into a youthful body. Everything's okay. I think 20 years is extremely ambitious for that. That's just me.
Your mention of CRISPR reminds me, I'm halfway through Walter Isaacson's biography of Jennifer Doudna, and it is excellent. I highly recommend it. I learned a lot. I'm listening to the Audible version. I'm about halfway done, and there's a lot of science in there. It's a really well-written book.
Very good. Do they talk about why? Because I know Carpentier and Doudna got the Nobel Prize. Did they mention why there is no other person on there? Because you can get 3 people a Nobel Prize.
They did not mention specifically how the Nobel Committee made their decision. No.
Just curious. Okay. Anybody have any other cool insights into this paper? I think we've traversed a lot of ground here. I'm super excited about this technology. You know, now that it's at least shown, you know, we've shown quite a high level of safety in mice. So that's always a huge deal, you know, for preclinical trials, so to speak, before you end up putting this into patients, right? Still some years away from putting it into human patients, but yeah, I definitely see this, you know, being studied and applied to organ regeneration. And there might be a follow-up paper where it's not aging per se, because I know we looked at a paper which looked at a different clinical point, idiopathic pulmonary fibrosis, and the treatment thereof, using Physidin and I think other, it might not have been Physidin, it might have been other senolytics, right? And that was a treatment, that was a disease that that is in search of a treatment for humans, but it's not aging per se, right? So I wouldn't be surprised if these OSKM factors, using this methodology, would be used as a kind of a more focused intervention to get a, tissue regeneration and a reversion to a normal, healthy physiologic state for a individual organ, for example, such as acute kidney failure or something like that. So I think we're going to see a lot of papers in that direction, stemming from the work from this paper.
Excellent paper.
Yeah. Cool. So anybody else? Any comments? Any thoughts, suggestions?
Yeah, so just consider the future. So we've got something a bit different next time, have we not?
Yeah, I believe we will. So we will be. Well, we haven't picked a date exactly, but we might be having a joint session, or planning on having a joint session with folks from an organization called Vita DAO, where we might be going over a. Well, the plan is to go over a theory paper. That was the. Keep forgetting, Steve. What's the name of the paper? A hyperfunction.
Yeah, it relates to the hyperfunction theory of aging and the developmental theory of aging by Pedro. But yes, it's a paper by David Gems, and it's quite interesting. Well, I think so, and I've been right before, so
yeah. Well, of course you have. That's why you're here.
But yeah, I honestly think it's quite interesting because we've talked about this before, that the models that we've got, the hypotheses of aging, that none of them really quite sort of fit the bill. They're all sort of right in some ways and wrong in others. And we've talked about moving towards that grand unified theory of aging, haven't we? And I honestly think this paper, does a good job at sort of advancing our understanding of aging. And I think that's very important, that we need to get that understanding of, I wouldn't say everything, because there's a lot to work out, but I would say that we need to know more than we currently know, should we say that? So I think this paper does a good job of that, and hopefully you'll share my enthusiasm for it. And it's got something for you programmers and you damage theory of aging sort of proponents, because it's both, which is what some of us have been saying for bloody years, which is it's not program, it's not damage, it's probably both. And let's talk about it. Let's share some views. I know Oliver will have something to say, but I honestly, I think the programmatic stuff in that paper may well actually not trigger you.
Yeah, I want to get who I want to who want to get online. I want to talk to Leonard Hayflick. I got to get Hayflick online. I know he's probably in a state of like he's retired, but he's he certainly wrote a he certainly has his views on programmed versus non-programmed, and some of it, and we have to, and I think what we can do when we have this discussion on our external club is try to make sure that we're all talking about the same thing, right? When we talk about, and I don't want to, this meeting we should be closing it soon, so I don't want to talk too much about theory right now, but but we should make sure that we're clear as to what we're talking about, because sometimes we can get into arguments about, no, this is programmed or it's non-programmed or it's both, but we could be very clear as to what we mean by what's programmed, right? Certainly that something is programmed and accelerates aging doesn't, to me, in my mind, doesn't necessarily mean that aging is programmed. evolution is a complex thing, right? That's where you end up with having antagonistic pleiotropy, right? Something, something you need to fundamentally not be eaten by something out in the wild and propagate your genes. So if you run faster and that puts more strain on your body because the environment is harsher, versus not running so fast and living longer, well, you won't live long enough to procreate if you get eaten, right? So you might need to put some wear and tear on your repair systems for immediate gain, so to speak. So so there's that, and I don't want to. We can unpack that later, but certainly I veer definitely towards that, but that's not. I don't think that's what I would consider to be programmed aging. I've read quite a few books and papers from people who talk about aging as if it didn't exist at some point in the past and had to evolve later on when during in cells that were multicellular or sexual. And this was like an immunological book I had read a few years back that was written in the late 90s, and there's so many things that since then have kind of proven wrong, where where we do have studies showing that individual microbes do age, right, as long as you can track them very carefully. And you have to be very careful and nuanced about individuals versus populations of individuals, right? So individuals will age, but populations won't. And then we get into the nuances of what is a population, right? So you can talk about, well, certain multicellular organisms like Hydra don't age, although they're undergo a lot of mitosis, and there's a lot of cellular turnaround, and there's a lot of also cellular plasticity in a Hydra, in that different tissues can basically switch from a differentiated state to an undifferentiated state, right? So certain organisms, the concept of individual and population gets a little bit fuzzy, right? So versus a complete dichotomy of one extreme, such as ourselves, where you have somatic cells versus germ cells, right? A lot of organisms that are somewhere in between, and things kind of can switch back and forth. So yeah. So you have to, I think when when these discussions are made, when people talk about aging and longevity, to keep in mind these distinctions and these nuances, and so that we're talking about the same thing, because then otherwise we're all arguing different points and not knowing it, right? So anyway, that's just my 2 cents, and maybe we'll elaborate on that further when we talk more theory, right, and less application, as we've been doing. We've been looking at some hardcore experimental application papers these past several months. So let's go back to theory. Let's ground ourselves. We haven't done some theory papers in a long time, and theorize, because it's fun to theorize. Somebody once said, I forgot who it was, that like everybody doubts everybody is suspicious and doubts theories except the person who came up with the theory, and everybody is accepting of experimental data except for the person who did the experiments, right? Right. So there's a bit of a truism there, right? So anyway, that's all. I got nothing else to add. I think I'm done with this paper for today. It was fantastic, and we will have another, I think, great session next time around.
Well, we will do. So it sounds like you need to establish the ground rules at the start of what we're really talking about next time, so we're all clear. And I'm actually looking forward to it. So anyway, no more spoilers on that. We'll dive into some theory and see if we can solve aging next month. And that's about it. So thanks to everybody who joined us today. Quite a few people in today, and we will be, back next month, end of next month, around the same time. It's usually a Tuesday, last Tuesday of every month, and we'll talk some truly deep geeky stuff next time. So thanks very much for joining us, and especially to our heroes who make this possible. Thank you very much, everybody.
Thank you.
And if you're interested in supporting this and all the many other wonderful things that we do, and we do a lot of stuff, check us out. Visit lifespan.io/heroes and find out how you can help us do more. So on that note, I will bid you adieu.
Take care, everybody.
Till next time.